All right, well, why don't we go ahead and get started? I am Frank Kosh. I'm the Communications Officer for the International Pest Risk Research Group. And this is, I think, the 12th or 13th. We're going to say 12th. I'm not superstitious, but we'll go with 12th. This is the 12th webinar in our series. And today's presenter is, my Polish is bad, but I'm going to try. It's Dr. Anna Szyniszewska. Is that close?
Close enough.
Close enough. Anna is now the technical lead on the Global Burden of Crop Loss Initiative at CABI. She's also a scientist and founder at Corvus Geostat in Poland. She completed her Master of Science and PhD in Geography at the University of Florida, working on estimating climatic shocks to rice production in Thailand and interdisciplinary spatial temporal factors affecting introduction pathways for Medfly into Florida and California. And that was for PhD stuff. She spent 5 years as a postdoc at Rothamsted Research and 3 years as a postdoc at the Plant Sciences Department at the University of Cambridge. And she has experience working on highly collaborative projects tackling crop pest problems in the USA, Europe, and sub-Saharan Africa, modeling spatial heterogeneity in the distribution of host plants, monitoring the spread of pathogens, advising surveillance strategies, working with qualitative farmer survey data, modeling pest climatic niche and pest population dynamics. And her presentation today is Fruit Flies in a Warming World, Models and Evidence of Range Expansion. Before Anna starts presenting, I'll remind everyone, and it looks like everyone already has, but please do mute yourselves. You can leave your cameras on or off. I don't have a strong preference, but in any case, if you have questions, if you would, type them in the chat. We should have time at the end to do sort of a live question and answer, but it's often good to prime that by putting them in the chat. And I'll monitor what's going on in the chat for questions. Other than that, I want to remind everyone that we are recording this and the recording will be available afterward. I will say that again at the end and give a couple of announcements, but let's go ahead and say, Anna, you can go ahead and take it away.
Thank you, Frank. Thank you for the introduction, and thank you for inviting me to be part of the IPRRG webinar series. So my talk today will focus on climatic niche models using CLIMEX to illustrate how it can reveal spatial trends in species range expansion in a warming world. While multiple papers seen recently was receiving for review that explore climate changes scenarios, typically from 2030 onwards, I would like to put in this presentation a magnifier glass on more recent history and explore the last 50 years and the trends that we are observing that are related to the expansion of those fruit flies that I'm going to talk about. Those recent trends in climate changes, they have implications for modeling species climatic niche and also especially interpreting recent species occurrence data, which sometimes are a little bit tricky to distinguish whether these are ephemeral population or established or maybe newly established populations. So I would like in this presentation raise attention to this. And often I try to cross slides as slow, but okay. So this work was part of FFIPM project. It's a European research project funded to protect fruit production and address trade threats posed by fruit flies in Europe. The objective of the FFIPM project is to establish in silico supported prevention, detection, and IPM approaches for 3 fruit fly species. One of them is Medfly, Ceratitis capitata, which is established in Southern Europe, but we are observing increased occurrence of this species, so outside of what we consider to be its established zone. And also two other fruit flies, one of them oriental fruit fly, Bactrocera dorsalis, and peach fruit fly, Bactrocera zonata, which are currently not established in Europe, but are serious invaders, and there is a likelihood they could become established. So the work on the project is based on spatial modeling, population dynamics modeling of those fruit fly, also designing real-time decision support systems for farmers, and filling gaps in knowledge, identifying the knowledge gaps related to those fruit flies biology, and developing experiments to improve our knowledge. As part of the project, my role was to explore what is happening in Europe and worldwide as part of global change. So change in climate and also change in the transport flow volume. We looked how does it affect the risk of fruit fly importation and establishment. And in one of our case studies, we are focusing on seasonal range dynamics of fruit flies and short-lived occurrences that may or may not be able to overwinter in invaded areas. But what I would like to focus on in this presentation is the climate change signal in the recent decades. Because I will not be showing work related to the transport, we'll be focusing on climate change signal. From those three species, I chose to show information about Medfly today. I will be also showing information related to Queensland Fruit Fly in Australia. So a few words about the global change and how it impacts the fruit fly range expansion. There were many studies recently showing how in a warming world, the species distribution in general, we are observing the poleward movement of species distribution. What it means is that the marginal areas of species distribution are likely to become increasingly suitable. And we may be observing overwintering of organisms that maybe a few decades ago were not able to overwinter in those locations. It also means that suitable areas may be able to accommodate more generations of pests in one season. And this is especially the case in temperate areas, but not only. As part of global change, obviously, we also see faster transport pathways. Commodities are being shipped faster over longer distances, and we are observing increasing volume of shipments of commodities and also movement of people. The movement of people, obviously, last 2 years would be a big outlier to the trend, but I don't think I need to convince anyone that overall that's what we see worldwide. And that obviously creates increased opportunities of establishment of new species in new locations and increased propagator pressure. Also, the proliferation of irrigation areas in drier climates, they increase the areas where, like the source areas of potential sources from where species can be transported. And that's definitely the case for fruit flies and more areas that are suitable for example, oriental fruit fly or peach fruit fly in drier areas south of Europe. So I will focus first. I will just give a brief description of two fruit flies that will be the topics of my talk today. First one is Mediterranean fruit fly. It's a very invasive species, which is affecting predominantly tropical and subtropical areas. It's originated in Africa, but it has consequently spread from its origin, most likely in Kenya, all over the continent, and it is present currently in North Africa, Middle East, and also Southern Europe. It's also present in Caribbean, Central and South America. It is established in Hawaii, and there are frequent observations of Medfly in California, and also frequent interceptions in Florida. It is one of the most economically important pest species. It's highly polyphagous. It can feed on over 100 different plant species, especially on citruses, peach, apples, and mango, but there are many more host plants that fruit fly likes to feed on. Oops, and often scrolling my slides in Zoom is not very responsive, but here we are. And second fruit fly I will talk about is Bactrocera Triony, Queensland fruit fly. It is the most serious insect pest of fruit and vegetable crops in Australia. It has very wide climatic tolerance and as well very wide range of hosts. It infests all commercial fruit crops in Australia other than pineapple. It is found throughout the eastern half of Queensland, eastern New South Wales, and east of Victoria. It has high invasive potential, and of particular concern are now repeated incursions into New Zealand, so South Australia, and the first ever incursion into Tasmania. So, apparently, like, those populations have not become established in these territories, but it's a big concern they could. So as mentioned at the beginning, I used CLIMEX to model the climatic niche of those two fruit flies. CLIMEX, it's a hybrid inductive and deductive model, and what it means, the predictions rely on biological parameters related to optimal temperatures or optimal moisture thresholds, and also the maximum and minimum thresholds for moisture and temperature below or above which the growth of a species is not occurring. And the model, I'm sure many listeners today are well familiar with CLIMEX. It allows this cross-validation between using information derived from experimental studies and verifying it against species occurrence data. So it's a very iterative process to look from what we know from the literature and verifying it with what the model predicts. It simulates geographical and seasonal climatic suitability, length of growing season, number of generations. It incorporates also climatic stresses above or below which the species starts to accumulate sort of negative growth, and at some point these stresses can become lethal. And you can apply irrigation scenarios in that model, and it has ecologically consistent response to novel climate scenarios, those models. So the outputs of the models, like some of the most important outputs and informative for the user to understand what processes are happening that determine whether or not the area is suitable for the fruit flies. So the most important ones, one is the growth index and another one is the ecoclimatic index, and they range from 0 to 100. They illustrate the overall climatic favorability for the species at specific location. And the difference between GI and EI is that GI represents the potential for population growth and development without considering stresses that may inhibit or potentially completely prohibit population growth. So there is an opportunity in CLIMEX to explore the temperature index and moisture index, and those indices, they represent response of the species to daily temperature cycle or the availability of moisture in the soil and how favorable it is in a given location. Stress indices, as I mentioned a moment ago, they are accounted in calculating the EI. So even sometimes if the growth index is very favorable, the stress indices, if they accumulate to values above 100, they can go up to nearly 1,000. The high stresses, for example, cold stresses, dry stresses, or wet stresses, or heat stresses, they can prohibit the population growth and may reduce the effect of GI and may result on very low ecoclimatic index. And the ecoclimatic index of 0 will indicate that area is not suitable. Regarding the parameters that I used for both models, for QFly, I used the parameters from Ionov and Sader's paper from 1998. I've done work on fitting new parameters for the Medfly CLIMEX model to update the model that was published by Teresa Vera in 2004 or 2006. For the climate data, I use a time series, which was actually from 1901 to 2019, but I used climate data for 1970 to 2090. That's from the Climate Research Unit, TS4.05, at 30 minutes spatial resolution. I also used the Climont 30 dataset for centered around 95 for like a more general prediction, but this dataset is not enabling. It's looking at the 30 years averages. Climatology doesn't allow time series exploration of data. I used also for the irrigation mask from FAO GMIA to classify areas as irrigated. So we classify them as irrigated if they had 10 or above 10 hectares of irrigation per pixel. And I ran the model using two scenarios, no irrigation and with top-up irrigation of 2.5 millimeters a day. For the project, I conducted with my colleagues and also more partners on the project, conducted very thorough literature search for reported temperature or moisture thresholds defining optimum, maximum, minimum values for Medfly survival. And there was a very wide range of reported results for different. We classified those results based on the stage of the development, so eggs, larvae, pupae, or adults. And this information was guiding our choice of parameters for the CLIMEX model. This is the choice of the parameters. We have parameters from the Ionov and Sader's paper for QFly, and we updated the Vera paper for Medfly. We then ran the model in two irrigation scenarios and produced composite maps. So in dry areas, as we know, the development on fruit flies will be very limited by moisture availability. So some of the results I will be showing are from California, and most of the results in California will be an effect of this irrigation scenario. So assuming that we have top-up irrigation in those areas. And so the results are an ensemble of two models where we, in any area that this mask is indicating as irrigated, we are showing results from the irrigation scenario. In any areas that do not indicate irrigation presence, the results will be from the scenarios which did not consider top-up irrigation. So this is explained on this figure, which was borrowed from Tanya Ionov paper from 2016. So in A, here we have shaded areas that are suitable under a natural rainfall scenario. Here in B, there are shaded areas that are suitable under an irrigation scenario. And in C, we have the hatched areas that are irrigation areas. So in result, we take a portion of pixels from B within the irrigated areas, and the remaining ones will be from model which is not assuming irrigation. So the first results that I'm going to show are going to relate to the Medfly model. We conducted a very thorough search for Medfly occurrence data. This is a result of a quite filtered information. There were many more occurrence data which had to be heavily cleaned. Portion of this data comes from the Global Biodiversity Information Facility, which I'm sure many of you know and use for occurrence data, and probably you also come across challenges that it's a very prolific database, but actually needs a very careful approach and a lot of cleaning and removing points that may not be relevant. Also, our collaborator, Mark de Meyer, helped us with shared his records from the Royal Museum of Central Africa. I used portion of information that I gathered during my PhD work, where I was browsing the published literature for additional Medfly occurrence records. We sought expert opinion on outliers. With this big number of occurrence points, it was very important to gather information which are unlikely, which can be erroneous. Records dated pre-1950 were removed. I don't only because it's often unclear what do they like, whether or not they represent the actual location of occurrence, or they may be associated, like geocoding. It's more dubious in those records. And we reviewed data for cleared outliers with incorrectly typed coordinates and those which evidently were showing country or administrative unit centroids. We removed them from the data set. For me, the biggest challenge was actually there were many records from Europe, and we also sought expert opinion, which of them represent established populations and which of them represent ephemeral populations, or like completely like only short periodic occurrence information in areas where they definitely cannot overwinter. So for example, in Romania, in 2013, there was a paper published indicating that like Medfly locations in Bucharest, in Constantia. So we were curious what the CLIMEX model will show, whether we will see suitability or increasingly suitable climate in those locations, because it did seem like quite far off from what we know that it's an area of Medfly establishment. There are frequent occurrences of Medfly around Vienna, in Australia. And also, as we know, frequent occurrences in the Paris Basin. So also we were wondering, is it something happening also in Paris Basin? Is the range going this far north? Can we at some point expect that Medfly may at some point become established in this area? And also these locations maybe around Lyon, in the Rhone Valley, they seem quite close to the area which we know that where Medfly can overwinter. We were also wondering, is it something that is indicating more persistent populations or not? And actually in Italy, it was also a very interesting case, because there are reports of overwintering of Medfly in the northern area, so in the Po Valley and even in the Trento region. So there are some published papers which indicate that there are very small populations that are apparently overwintering as larvae. So when we were gathering data, those points were not described. They didn't have any indication whether or not it is established point. So it was a lot of for this data to serve as a guide. It was not very clear, and we had to seek a lot of expert opinion which points represent established populations, which not, but we did not get a clear consensus on many of them. So now I will start showing the results of our model, and I will be showing them as a, so here I'm showing the decade between 1970 and 1979, the average ecoclimatic index using our model parameters, average growth index, temperature index, moisture index, cold stress, and heat stresses. So heat stresses in general in Europe are 0. This is not a big concern. The cold stresses is the biggest limiting factor of the Medfly potential for establishment in Europe. And this is the picture from between 2010 and 2009. So when I sort of scroll between these two decades, we can see that the EI is expanding northward, the GI, TI, they are all having higher score, like in more northern locations. And the same is the case for cold stress, that it's actually, we have darker areas in Southern Europe because the cold stress is diminishing in Southern Europe. So we looked at the time series of 50 years, and for each location, for each pixel location in Europe, we fitted a linear trend line for the index values, so ecoclimatic index and the rest of indices, as well as cold stresses. And we looked what's the slope, what's the annual rate of change in these indices in those locations. So here we have the trend parameters. So 0.5 means that every year we'll expect, so in every 2 years, we will expect a change by 1 point of ecoclimatic index. So in 50 years, it can accumulate to significant change in some locations. So for example, in locations with the highest range, it can accumulate up to, well, maybe 20, 25 even, change in the ecoclimatics index values. And just to illustrate that, this is the linear trend for Marseille in France. So we can see that the values are increasing from above 12 to nearly 25. And for the ecoclimatic index, area above, like anything above 1 really indicates it's suitable, although 1 may not be very suitable, but I would say everything above 5 will indicate that very likely, like the model is indicating that the species is likely to persist in that location. So we looked at the trends, and we also looked at the p-values. Many of those trends were not significant, but in all bright yellow colors, you can see in which areas the trends were statistically significant. And we can see that in Southern Europe, many areas indicated significant positive trend in EI values, GI values, the thermal index, and also this will be actually here a negative trend in the cold stress values. So this map is showing the areas with significant trend changes. So we masked out all the areas where the trends were not significant to indicate which areas and what was the rate of increase. And we do observe increased suitability over the last 50 years. So the trend that is continuing to rise, and which can imply that we are very likely to see Medfly range expansion in Europe. And for each of those locations, we examine what is the trend. So for example, the locations in Romania, it was very, very difficult. Like we would need to use biologically implausible parameters in order to make southern Romania suitable. So we were wondering, is it an effect of, it does seem from our model that it's very unlikely these areas could support long-term establishment, like above a few seasons. The same in Austria. Does seem that, but we see this area of southern France and also northern Italy becoming more suitable. The same is the case in Spain and Greece. And we can plot this information for every individual location to understand those trends a bit better. And this illustrates the difference between EI mean values between 1979 and in 2019. So what was the average ecoclimatics index value in Europe, and what were the absolute differences between those values? So we can see again that we see significant increase in suitability. And this, finally, this map for Europe and Medfly indicates in yellow that based on our model and the parameters we chosen, which areas became suitable when we compared the first and last decade that we looked at in our study. And this area in northern Italy, as also confirmed by some recent studies, does seem to become more suitable. And these plots indicate values for California. They represent the ecoclimatic index value, growth index value. So similarly, the same pattern here of plots and observations as we had in case of Europe. And this represents the composite model, so it does account for irrigation. And as I said, in California, the availability of moisture is a very limiting factor. And also it illustrates the effect of cold stress in Northern California and in higher areas of California, how it is the difference between the first and last decade that we looked at in our study. This map represents the areas with significant positive trends of suitability for Medfly, although I must say here the increase in suitability it's not as steep as we were observing that in Europe. The scale is like it has different values here, so but we also observe increasing trend in Medfly suitability and decreasing trends in cold stress values in California. And this is a map representing the absolute differences between ecoclimatic index values derived from our model for California. And in California, so as I said, the trends, it can be seen here on the on these plots, that while many of them are significant, however, this is quite wide scale here between 0 and 100, but this rate of increase is much gentler. However, looking at 2 decades, we observe the potential northward expansion of areas that are potentially suitable for Medfly. Right, and the second fruit fly I would like to illustrate in this presentation is the Queensland Fruit Fly, very significant pest of fruits and vegetables in Australia. We applied the same methodology using the parameters from the paper published by Jonov and Saderst. This is obviously Southern Hemisphere. And again, we looked like between the 2 decades how those indices and their values were changing in Australia. We also observe in this area here in New South Wales and southwest area of Australia, increasing suitability for Q-fly as well. And we also see that the moisture index value are becoming also less favorable, that can impact the suitability in other areas. And this plot is showing the absolute differences, and we see quite pronounced increase in suitability in that area of Australia where actually Q-fly is established. But the topography in Australia, like here we have higher areas, so we see this southward expansion of areas that are potentially suitable. We don't see the suitability in Tasmania or in Southern Island in New Zealand. This is here limited by the topography, but it does seem that the suitable areas are expanding. And we can see here negative trend in northern areas of Australia. However, gentle positive, like increasingly suitable areas in like very south of Australia based on the model. So the conclusions are that we are observing like the last 50 years brought a lot of changes. So the climate is steadily warming, and it does have implications for modeling and understanding the species climatic niche. I see many papers which still look at species climatic niche using data centered around 75, which mean it looks at climate average between 1960 and 1990s. And this is very outdated. I would say like it should not be represented as the current climate. Climate, unfortunately, is changing. It's becoming very obvious looking, like examining those models. So we sort of need to consider new climate data, most recent climate data in those studies in order to represent correctly the current distribution of species. So persistence of Medfly in Romania and around Lyon would require biologically implausible parameters. When we were fitting the parameters for Medfly, it would require significantly decreasing the thresholds for the cold stresses. It didn't seem like it's likely that the populations can persist there. Persistence of Medfly in northern Italy and Paris Basin or near Paris Basin is becoming increasingly likely. The biodiversity databases, they increasingly are prolific with casual species occurrence data, and it is for the users, it's making it quite difficult to distinguish which points are representing established populations, which not. it would be nice if we had a bit more information and classifying this information in those databases. And also occurrence records are often also in those databases are not dated, and that has also implications for species niche modeling. Right, and I think, yeah, and that's it. I think that's the last conclusion point. It came here a bit by accident, but thank you very much for your attention, and I would like to now open the floor to questions.
Great. Thank you, Anna. Yes, so we are open for questions. If you want to, yeah, well, if anybody wants to ask a question, I guess you could get started. But if you don't mind, Anna, I have a couple questions related to, well, first I have a comment. I agree with you that there are still a lot of people looking at an outdated climate, looking at that centered on 75. And yes, preferably everyone should be looking at a more modern normal. I don't know how we make people do it other than encourage people to use the data that are out there. But that's just one comment. My question is more around the occurrence data. I agree with you again that there is a lot of casual observations that are in there. So my question is, so thinking about the Medfly in particular, do you have a rough guess as to how much you had, what percentage of your occurrence data did you end up having to omit? And it could be for Europe, or if you want to talk about sort of the global occurrence data set, that's fine too. You have a guess on how much you had to get rid of?
I think probably about, I would say, if I had to give, I can actually look back and exactly calculate. I would say probably about 30% definitely had to be discarded. And many of those points, I must say, I was aware that they represent just outbreaks that were controlled, or populations that were not established. So in some cases it was easy to distinguish. It also depended on the source where we received the information, but there is a big chunk of information that usually needs to be discarded when we work with those big databases. And for example, Europe has plenty of records in Scandinavia for Medfly, in Scandinavia, in the United Kingdom, Northern Germany. So areas which, like, we could say for certain that they could not represent overwintering populations, but it's not so easy to work with the database because they are often not classified as these casual interceptions. So you don't know if you are trying to filter it by attributes. You need to have some, and that's good probably, that you need to have a bit of knowledge, and but sometimes it's just getting harder and harder this tangle the closer you get to this areas, this marginal distribution areas, as that was the case for Europe. In northern Italy, central France, we were seeking expert opinion, what do they think, but often they also don't know for sure.
Yeah, I mean, it seems to me that maybe this is calling for, and maybe this exists, I'm not sure, but maybe this is calling for, you know, some guidelines, some guidance out there on how to, because you're right, these casual occurrences, or there's a lot of ways for those occurrences to end up in a database that any scientist or analyst is going to use. So maybe we do need some guidance on how to address that. Okay. All right. So if you have a question, you could type it in the chat. I don't see anything there right now. Or if you want, just sort of announce yourself, the floor is open, and we have certainly have a few more minutes for questions. Anyone else? Well, I tell you, I have another question, or I have another sort of point to work around, and that's the irrigated areas. And I was thinking about the irrigated areas. The California example, to me, that's a good one, because when you think about the places that were suitable was mostly the Central Valley, which of course is a heavily irrigated part. And you know, I'm just sort of wondering, like, if you're tracking backwards and you're looking at that first decade, is that also assume, is that sort of, are you, I guess you're assuming that the irrigated areas are fairly consistent through time, like going backwards, like where they're irrigated in recent history is the same as where they were irrigated, say, 50 years ago?
Yes, yeah, it's far from ideal, the irrigation mask, for many reasons, as you say, because this is also a changing, it's changing, and we have only one snapshot. I know there are efforts to improve those irrigation layers, to look at satellite imageries to determine where we see the biomass that would exceed what we would expect given the precipitation. So there, I look forward to a better irrigation masks, and you know, in many areas there might be irrigation, but it may last for some time, but those sources of additional water may also become depleted with time. So the assumptions that this irrigation will be available constantly throughout the year, it's also a gross assumption to make. Yes, Frank, it could be better, and I look forward to.
I think, you know, I when people work on those developing the irrigated areas, I wonder if you could use time series from remotely sensed data to sort of estimate how irrigated areas might have changed through time. But you know, that's a big, somebody could devote a big chunk of their career just to working on that. So
but I hope we'll get there. I think definitely there are many opportunities.
All right, so we do have a few questions, and I'm just going to read, if you guys don't mind, I'm just going to read these off. So the first one is from James Carey, and this is, would your model predict the confirmed temperate distribution of the tropical, subtropical species of Drosophila, such as D. melanogaster, as a test of Medfly outliers? So was that clear?
James, could you maybe elaborate?
Yeah, if you don't mind, why don't you go ahead and turn your, if you can go. Yeah, here we go.
Yeah, sure. So, you know, to use data where you have Drosophila, which is essentially a subtropical species from Africa, Asia, that would be a test to, you know, of your model to see if it actually predicts a subtropical species occurring in some of the more northern areas. And that would presumably give you some assurance that, or not, on the correct call on some of these tropical tephritters.
Mm-hmm. Yeah, I would be happy to look into that. Yeah, I cannot say now whether I will need to look at data. Sorry, I'm not that familiar.
Well, maybe you could comment. Maybe you could comment on the concept. That would this be some way, a method for validating your calls on the predictions for some of these tephritters?
I don't see a reason why not. I think it's definitely very possible, but I would need to see a bit more we need to read about the topic a bit more, James.
Okay, thank you.
For sure.
Okay, we have a couple more questions. This is from Dominic. Are those climate data sets open access and ready to use in CLIMEX? And so maybe Dominic's asking,
Yeah.
what did you use Climond, or I'm going to stop. I'll let you answer that question.
Yes, so Climond dataset centered around 1995 is definitely available on the Climond website for CLIMEX users. But for some reason, I still see people publishing models using Climond database centered around 75. So 95 is available. The CRU data that I used, I'm actually not sure. It is also available. I will double check which source did I use to receive it. So I would be happy if you'd like to email me. We'll be happy to point you to the source.
Yeah, that's and we have sort of a follow-up question on that. This is from Patricia. I think I'm being naive, but is there a new source of data or a source of newer climate data? So for example, is NOAA putting anything together for the United States? Is there anything for the EU? And I put in the chat, and if everyone will go take a look, I put in the Climond URL, but so I guess that's the short answer, right? Is go to Climond, there is newer. I don't know how much we have anything centered on, say, 2005 yet, but
yeah. So there are actually, as we all know, there's the climate data sets are very prolific, but it's a question of having the right variables and the right format for if you are working with CLIMEX or if you work with another tool for your tool. Yeah, so Climond is they have the source for the compare locations function, but the compare years and locations function, which looks at the time series data at annual time series or weekly time series. I'm sorry, but I don't recall which website did I use to extract this information, but this is a CRU dataset that I used in this, so the climate research unit. And this is for the globe, not only for Europe. This is data for the entire globe at half-degree resolution.
We have a couple comments from Ross about that. So Ross says, Anna, job well done. This is very interesting. I appreciate your use of irrigation classification, and the time series results are really compelling. Both in terms of your results as well as convincing researchers to make use of the most recent climate normals data. So thank you for presenting your research. And then later, Ross says, I just checked, it appears the Climond data set for CX is centered on 1975.
Yeah. Yes, but there is if you scroll down, there is one more data set. It should have information centered around 95. And if not, if this doesn't work, please email Climond, but this data should be there on the website, and I looked at it, I think, a few days ago.
Okay. Well, since we're almost at the top of the hour, there is one more comment, and I'm going to get this so we can fit this in. And if we go over a couple of minutes, I think that's fine. Hi, Anna, could you please, this is from user APZSHEN. So hi, Anna, could you please describe briefly how you do statistics on increased suitability? By the way, you're looking into decreased suitability is very interesting, kind of serves as a control against other factors that may help to increase the distribution.
Yeah. So I used R to analyze the outputs of the CLIMEX model, And so I imported everything into R. And for each pixel, each individual pixel, I looked at the time series for that pixel. So in that particular location, I fit a linear model function. So just general linear model and fitted function to it and extracted the information about the slope parameter and extracted information whether or not this slope, what was the p-value of that slope parameter. So that the p-values I used as a mask, so all the p-values that were below 0.05 I considered to be statistically significant, and for those areas where this we had significance, I illustrated what was the slope parameter value, which indicates the annual trend in change. And for some areas, indeed, it's positive. As per the trend is positive. In some areas, we are observing decreasing suitability. I also illustrated the indices value because because this can be different factors that drive that increased suitability. As for the case of Medfly in Europe, that was the temperature index and the cold stress. So increasing temperature index, so increased favorability, warmer climate, but also decreasing cold stress, because cold stress is the main limiting factor of Medfly distribution in Europe. And I see that
so Anna, if anybody wants to follow up, can I drop your email address in the chat right now? I mean, I'm sure they could find you other ways, but do you mind if I just drop your email address into the?
Yes, no, I don't mind. I can also put it.
If you would, I'm gonna close out because yes, if you would drop it in the, there we go. Great. Yeah, so we're just past the top of the hour, so I'm gonna go ahead and say thanks to everyone for attending. Thank you so much, Anna, for presenting and presenting on a reschedule. I appreciate your flexibility. It was very good. Everyone, this recording will be available. Hopefully next week I will have it up. And I want to remind everyone also that we are planning, if at all possible, to have an in-person meeting of IPRRG in Athens, Greece in mid-October. It's the week 10th through 13th of October. So I will be sharing more information about that. Also announcing when this recording is available. I will share that with you if you monitor the Facebook page, our Facebook feed, or our Twitter feed. I will announce those things there, so you will see a lot more about the meeting. And again, thank you so much, Anna. And I'm going to go ahead and stop the recording.
Thank you, everyone.
Enjoys rest. Yes. Do you have anything you want to say, Anna, before I hit stop?
Thank you for this opportunity, for inviting me, and thanks for everyone who connected today.
Oh, the pleasure was all ours. Thank you again. All right, so long, everyone. Our next IPRRG, we currently are scheduled for a webinar on, I want to say coming up in mid-April, and I'll announce that too. Just keep an eye out for emails and on our Facebook and Twitter. All right, thanks again.
Thank you. Bye.